My name is Woohyun Kwen, and my project is about diabetic retinopathy, which is a diabetic complication of the eye. 10.5% of the US population is diagnosed with diabetes, and this number will rise to 33.3% by 2050. Several people are at risk for diabetic retinopathy, a leading cause of blindness. This complication is caused by alterations in the blood-retinal barrier. The blood-retinal barrier preserves an optimal environment for vision. It consists of astrocytes, which maintain the vascular integrity, and signaling proteins called cytokines to photoreceptors. Photoreceptors maintain the visual function of the retina, and endothelial cells form blood vessels. While I was wondering how the blood-retinal barrier is altered in diabetic patients, I found it very interesting that patients with uncontrolled blood glucose levels are more likely to develop diabetic retinopathy than patients with normal controlled levels. In addition, astrocytes treated with high glucose have decreased ability to maintain vascular integrity. However, the exact mechanism of how high glucose affects the blood-retinal barrier remains obscure. In order to understand the effect of diabetic conditions on the barrier, I have investigated the role of high glucose-mediated astrogliosis on photoreceptors of the retina. Astrogliosis occurs when astrocytes become reactive due to abnormal conditions, resulting in their secretion of excess inflammatory cytokines. I hypothesized that high glucose would induce astrogliosis. Due to inflammatory cytokines, I hypothesized that an inflammatory microenvironment would be induced for the photoreceptors, leading to their degeneration. In addition, since anti-TNFα therapy is sometimes used to treat retinal diseases, I hypothesized that TNFα would be an inflammatory cytokine that be overproduced by these reactive astrocytes. Upon further reading, I learned about microRNA 155's role in glucose metabolism in adipocytes. MicroRNAs are short, non-coding RNA sequences that inhibit gene expression. MicroRNA 155 is known to be an inhibitor of PPARγ, interestingly a gene recognized to control glucose metabolism. I have investigated the effect of this inhibitory mechanism of microRNA 155 on PPARγ in the photoreceptors in diabetic conditions. I hypothesized that the inflammatory cytokines would lead to the upregulation of microRNA 155 and the inhibition of PPARγ, further exacerbating the deleterious microenvironment for photoreceptors. I hypothesized that high glucose would induce astrogliosis, resulting in increased inflammatory cytokine production, and in particular TNFα secretion. This inflammatory cytokine will result in PPARγ inhibition through microRNA 155 upregulation, leading to an inflammatory microenvironment, ultimately resulting in photoreceptor death. An in vitro model of the diabetic retina was created with different glucose concentrations. These three concentrations were used, and 25.0 millimoles was set as the control. To induce astrogliosis, astrocytes were cultured in different glucose levels, and ELISAs were conducted for detecting biomarkers of astrogliosis. Then, the effect of astrogliosis on photoreceptors was modeled by treating photoreceptors with astrocyte secretome. qPCRs were conducted for microRNA expression, and ELISAs for protein expression. Astrogliosis was modeled by culturing astrocytes in different glucose concentrations. ELISAs were conducted to determine GFAP and TNFα protein expression, which are biomarkers of astrogliosis. When astrocytes were treated with high glucose, the protein expression of both biomarkers increased, suggesting astrogliosis. In addition, the appearance of narrow extended neurites suggests that high glucose induces reactive astrocyte phenotype. The results suggested that high glucose induces astrogliosis. Astrocyte and photoreceptor interaction in the blood-retinal barrier was modeled using astrocyte secretome. Secretome is a set of proteins secreted by the cell into its extracellular milieu. Secretome was collected from astrocyte cultures and used as treatment for the photoreceptors. Although astrocyte secretome originated from different glucose levels, additional biological replicates of glucose treatments ensured that the difference in glucose levels did not affect the results. It was hypothesized that inflammatory cytokines from reactive astrocytes, such as TNFα, would upregulate microRNA 155 expression in photoreceptors. qPCR results showed a 381-fold upregulation of microRNA 155 when photoreceptors were treated with reactive secretome. With the qPCR results, a correlation between increased TNFα levels and microRNA 155 upregulation was portrayed. Since microRNA 155 inhibits PPARγ in adipocytes, this relationship was investigated in photoreceptors. When photoreceptors were treated with reactive secretome, the ELISA suggested decreased PPARγ expression. Previously, microRNA 155 upregulation was shown in photoreceptors treated with reactive secretome. Results suggest that microRNA 155 upregulation correlates with decreased PPARγ expression in photoreceptors, and this relationship is induced by reactive astrocyte secretome. To determine whether decreased PPARγ expression correlates with an inflammatory microenvironment, IL-6 secretion, an inflammatory marker, was determined with ELISA. When photoreceptors were treated with reactive secretome, IL-6 secretion increased by 70%. The results suggest that inflammatory markers are increased when photoreceptors are treated with reactive secretome. Superoxide anion is another inflammatory marker. Mitochondrial superoxide anion assay suggested that when photoreceptors were treated with reactive secretome, they produced an increased amount of superoxide anion, indicating an inflammatory microenvironment. Lastly, an MTS assay suggested that photoreceptor proliferation decreases when treated with reactive astrocyte secretome. The decreased proliferation demonstrates photoreceptor dysregulation. When photoreceptors are treated with reactive secretome, microRNA 155 is upregulated, resulting in decreased PPARγ expression and glucose metabolism dysregulation. An inflammatory microenvironment is caused for the photoreceptors, which is suggested by increased IL-6 and superoxide anion production. The inflammatory microenvironment and decreased proliferation of the photoreceptors suggest their degeneration. After investigating the role of reactive astrocyte secretome in photoreceptor degeneration, I want to identify a specific molecule in the secretome. Since TNFα is already a target for retinal pharmaceuticals, and the data suggested that reactive secretome had increased TNFα levels, I decided to investigate the effect of exogenous TNFα on photoreceptor degeneration. Photoreceptors were treated with 50 picograms per ml of TNFα. The same assays that were conducted with secretome treatment were conducted. It was very interesting that the TNFα treatment suggested parallel results as the reactive secretome treatment. However, one notable difference was that compared to the 381-fold upregulation of microRNA with reactive secretome treatment, photoreceptors treated with TNFα only resulted in a 5-fold upregulation of microRNA 155. The significantly smaller fold change for the TNFα treatment suggests that there is another contributing factor in the reactive secretome that can be causing microRNA 155 upregulation. There are countless other molecules that upregulate microRNA 155 expression, and identifying these molecules is one of my potential ideas for my future research. Results from TNFα treatment on the photoreceptors paralleled the results from reactive astrocyte secretome treatment. However, the significant difference in the fold change of microRNA 155 expression suggests the presence of another molecule other than TNFα that upregulates microRNA 155. The results from my investigation suggest that high glucose induces astrogliosis, which causes the reactive astrocytes to produce an excess of cytokines, including TNFα. When photoreceptors are treated with the reactive astrocyte secretome containing TNFα, the upregulation of microRNA 155 results in the decreased expression of PPARγ. An inflammatory microenvironment ensues for photoreceptors, ultimately leading to photoreceptor degeneration. Photoreceptor degeneration results in alterations in the blood-retinal barrier, causing vision loss, absence of vascular integrity in the retina, and ultimately diabetic retinopathy. My study suggested a novel chemical pathway of the development of diabetic retinopathy. In addition, the significant difference in the fold change of microRNA 155 between TNFα and reactive secretome treatment photoreceptors suggests the presence of other molecules in addition to TNFα that causes microRNA 155 upregulation. Although current therapies for retinal diseases only target TNFα, more effective therapeutic approaches can be made by inhibiting other cytokines or molecules along with TNFα. In addition, microRNA 155's correlation to the inhibition of PPARγ protein expression in the photoreceptors suggests microRNA 155 as a potential biomarker for diabetic retinopathy. Lastly, the significance of astrogliosis on photoreceptor degeneration suggests astrocytes as a potential therapeutic target for diabetic retinopathy. However, my findings must be replicated in an in vivo environment as my research was carried out in vitro. This novel approach in prohibiting photoreceptor loss in the diabetic retina will enable diabetic patients to retain their visual functions.